Afternoon, everyone. Okay, so I'm going to be talking about depuration of bivalves, specifically oysters in this case. So depuration, just in case you aren't aware, also known as purification, is the main treatment process for Class B shellfish under European legislation. And essentially what we're doing is we're purifying shellfish in clean tanks or tanks of clean seawater, coming in various shapes and sizes. That picture at the top there is one example, but there are many. But they all work according to the same principle of recirculating seawater, usually via some sort of disinfection system, such as UV or perhaps ozone. It's a process that's intended for microbiological contamination, generally effective for bacteria, specifically E. coli, which is the statutory indicator that we use to show fecal contamination in shellfish. So it removes E. coli very quickly, very easily, but it doesn't work so well for viruses, and specifically norovirus, which is what causes nasty diarrhea and vomiting. And this is an issue that EFSA recognised in a report from 2012. Now, the problem is there isn't a standard in legislation for norovirus at the moment. So we have to rely on E. coli, the bacterial indicator, as a means of telling us whether we got safe shellfish or not. And what we do know is that depurated shellfish that are free from E. coli can still cause norovirus illness outbreaks. So E. coli isn't telling us the whole story, unfortunately. And as I say, norovirus is not normally life-threatening, although in extreme cases it has been known to cause death. But normally it's diarrhea and vomiting. Takes 2 or 3 days to clear. Pretty nasty though, and obviously it's not good for sales of shellfish. So it's not something we want to see in shellfish, and we need to remove it. So if you look at the graph here, hopefully you can see that this is a study we did previously looking at E. coli, which is the red lines. There's a straight continuous line and a dotted line. These are 2 temperatures we did at the time, 8 and 16 degrees. On the left-hand axis, we have here percentage organisms remaining, so 100% down to nothing at all. And along the bottom, we have number of days of depuration. Now you can see E. coli, the red line, is very quickly removed. 2 days, it's all gone. Now the green lines, again, we've got a continuous line, which is 8 degrees, and a dotted line, which is 16 degrees. You can see that even after 14 days, there's very little, if any, removal at all, perhaps a little bit at 16 degrees. If you contrast this with the yellow lines, this is a surrogate viral indicator that we use to represent norovirus in some situations. It's quite easy to grow. It's very similar to norovirus, so it occurs in sewage. And because of the difficulties in, you can't grow norovirus, so we use bacteriophage as an indicator to try and represent what norovirus might be doing in terms of infectivity. So with this particular study I'm talking about here, the green lines represent norovirus as determined by the molecular method, PCR. The yellow line, however, was the viability method. So we actually grew this FRNA bacteriophage. What we're going to be doing, or what we did in the Seafood Tomorrow trials, however, was to grow FRNA bacteriophage again, but also we did a PCR method. We developed a PCR method so that we could directly compare the results of FRNA bacteriophage, the viral indicator, with norovirus itself. So by doing both the PCR method for the FRNA phage and growing it as well, we're hoping to see whether, if there are any differences, we could relate those to what might be going on with norovirus. Because as I say, with the trials we did before, we just don't know how much of this norovirus is viable. I hope that's clear, but hopefully as I go through, you'll understand a bit more of what I'm talking about. So the aim of the study is to develop, obviously, enhanced depuration procedure based on norovirus removal and reduction in infectivity. And this is needed by the shellfish industry, particularly in the winter months. This is it's called winter vomiting disease. The peak norovirus season in temperate latitudes is in the winter months. So the current, under challenges, the current routine norovirus test method, as I mentioned, is a molecular one, PCR polymerase chain reaction, and it can't indicate live or infectious virus, and that's obviously the risk to the consumer. So we're using this surrogate virus, F-specific RNA bacteriophage. It can sometimes be called coliphage as well. I'll just refer to it as RNA phage, as it's a bit of a mouthful. We're going to use that as an indicator, or we used it as an indicator. Challenge is, can it be indicative of the infected norovirus risk? This is what we're investigating here. Now, the advantage of using FRNA phage is that it's easy to grow and quantify. So briefly, the methods. We concentrated on Pacific oysters because they are the main species of bivalve that is related to illness in consumers from consuming shellfish, generally because they're eaten raw or very lightly cooked. So Pacific oysters, Crassostria gigas. These were environmentally contaminated. We tried lab contamination, but it gives fairly inconsistent results. So for a number of reasons, we felt the environmental contamination was going to be more representative of natural contamination of shellfish. So we placed those near a sewage discharge with the help of one of our local operators. We tested various factors, water temperature, 18 degrees this time, rather than 16 for the previous study. 18 is around about the maximum that, from studies we've done before and colleagues elsewhere in the UK, that about the maximum that Pacific oysters can stand without getting too stressed. And 8 degrees, which is the other end of the scale, the minimum we tend to find where there's any useful activity. We also looked at feeding using microalgae that we grew in the lab and shellfish diet, which is a commercially available preparation. We looked at light and dark regimes, disturbance versus non-disturbance. And what I mean by disturbance here is vibration from the pump. Tanks often have a pump associated with them on the side of the tank, and that can cause disturbance, vibration transmitted through the water. The theory was that that might potentially upset the shellfish. So we compared disturbance, a tank with a pump, versus non-disturbance, so a tank without the pump, with water passed through from a sort of remote source. We also looked at continuous flow through of water versus recirculated unfiltered seawater to see if that made any difference. So we looked at various factors here. And then we tested the shellfish for norovirus, as I mentioned, by the molecular method, PCR, looking for genogroups, G1 and G2. They're the ones that most associated with human illness. And as I mentioned, we looked at FRNA phage by the culture method and also by the same sort of method that we use for norovirus, the PCR molecular method. And just to make sure that everything was moved as expected for E. coli, we looked at the E. coli as the statutory indicator as well. So the picture here shows the tanks that we use. These are commercially available tanks, tend to be quite popularly used in the UK, relatively small quantities, 90 kilograms a time. But the advantage is you can have a number of those, so you can scale up your business as sales increase. So we use commercially sized tanks for these trials. Now the results were interesting. We saw a difference between the two gene groups, gene group 1 and 2. Gene group 1, or G1, is you see the two lines, the continuous lines at the top there, the red and the blue. The red line is 18 degrees, the blue line is 8 degrees. So these are the temperature experiments. Very little removal, even after 5-6 days, very little removal of G1. But there was quite a lot, much better removal of G2, which is the dotted line, particularly at 16 degrees, 18 degrees, sorry. And if you look at the dashed lines, this was a bit disappointing because these represent the lines, the removal of FRNA phage, which is the indicator we were hoping to use as something representative of norovirus. Now, if it's going to be a good indicator, it's got to be less easily removed than the other pathogen we're looking at here. And as you can see from this graph, the FRNA phage was apparently better removed. Now, that may be because the levels we were looking at this particular time were a bit on the low side, because it, as luck would have it, or wouldn't have it in this case, the levels of norovirus and bacteriophage this particular winter's work weren't particularly high. So they were approaching the limit of detection for the test. So we may need to confirm this with further work. I should also say we didn't see much difference between the infectivity removal and the actual PCR signal, which does suggest that, going back to the initial graph I showed you, that the norovirus that was there, it may be that, that there's no difference between the infectivity and the viable virus at that particular point. So the conclusions from this study, Depuration is definitely better at 18 degrees than at 8 degrees. In fact, twice as good, we found, after 5 days. Now, there was a difference between the junior groups. Junior group 2 was much better removed than junior group 1. And the reasons for this may be due to binding. Certainly workers in Ifremer in France found that junior group 1 strains can actually bind to the oyster gut and be therefore much more difficult to remove. Whereas junior group 2 don't seem to bind to the same extent, and these were reduced up to 60% in our trial. So this does show that depuration can be useful for some strains of norovirus. G2 is certainly related to illness, linked to illness outbreaks, so it's useful to be able to remove that, if not removing junior group 1. Salinity needs to be matched to the natural environment. This is what we found from the results for salinity were less convincing than they were for temperature, but there did seem to be an effect suggesting that you need to match salinity closely to the natural environment, within 20%, say. Somewhat surprisingly, we found that the other factors trialled, including feeding, didn't have any significant approved effect at all. So that was a bit of a surprise, but so no apparent benefit from feeding the shellfish. As I mentioned, we did see that the FRNA phage was better removed in our study than both junior groups 1 and 2 of norovirus. But we do need to do further work on that just to clarify. In fact, we're going to be working with workers in France on a project called Oxivir 2 to investigate that. And finally, we found E. coli was consistently removed in all trials as expected. So summary outputs then. Elevated temperatures can lower norovirus levels from contaminated Class B oysters. This is assuming, of course, in temperate climates similar to the UK. Relatively cheap to add heating to systems, about €35 for the small system I showed you, 90 kilograms, maybe €450 or so for a bigger system, 750 kilograms. So our method can reduce health risks by improving seafood safety. Anyone is able to or can use this method. It's a straightforward method enough to use. We've produced a scientific paper in Food and Chemical Toxicology, peer-reviewed, reporting all this information. There's also a protocol that we produced for the industry for anyone to use if they feel like to. So all the information is there. Thank you very much for listening, and happy to take any questions.